Bacterial staining techniques. In this bacterial staining techniques chapters, we will discuss the various staining techniques or procedure for staining of bacteria. Staining, a technique used in microscopy to enhance the contrast in the microscopic image. That means staining will enhance the contrast of microscopic image. Mainly here we were discussing about the bacterial staining techniques or procedure so that we can study the morphology of bacteria. So we know that bacteria are microscopic organism because it is measured in micron or micrometer. That means with the help of microscope only we can see the bacteria. And without, if we are not staining the bacteria, it is very difficult to observe and study the morphological characters of bacteria. So what we will do before going for the for observing under microscope, we will stain the bacteria with different stains so that uh, bacteria will get some color and also it will enhance the, due to that color, it will enhance the contrast. So easily we can observe the and study the morphological characters of the bacteria. So staining is called as artificial coloring of cell or organism. With the help of this artificial coloring, it will enhance the contrast so we can identify and study the morphology of organism. So staining, we told that it is the artificial coloring of cells or microorganisms. The staining, with the help of this staining, we can observe the microscopic organism. So before doing the staining, we have two steps. First step is to the smear preparation. Smear preparation means what and all we have to observe. Mainly here we are discussing about the bacteria. So bacteria have to smear it on the slide, and after smearing it have to be heat fixed. Then only we will go for this staining. Smear preparation is a very important step before the bacterial staining procedure. Without the smear preparation, we cannot go for this bacterial staining. For the smear preparation, we required clean and neat glass slides, bacteriological loop, and flame. So first, we will take the liquid culture or sterile water with the help of this bacteriological loop, and we will place this liquid into the clean and neat glass slide. And after that, we will add the bacteria into this liquid and we will mix this liquid and bacteria properly. And after that, we will spread this mixture around 1 centimeter area in this slide. And after that, we will air dry or heat fix gently. When it dry briefly, the heat fix cell to the slide. That means after making the smear, we will heat fix the slide. Why we are heat fixing means after the heat fixation is over, the bacteria smear will bind to the slide properly. If we are not heat fixing the properly, the bacterial smear will be peeled off during the staining process. Type of staining. Type of stainings are simple stain, differential staining, and special staining. In simple stain, we use a single stain or dye to color the cells or organism. With the help of this simple staining, we can study the shape and arrangement of the bacteria or cells. In differential staining, after the staining process is over, we can differentiate bacteria into two. Either it is positive or negative like that. For example, the Gram staining and Ziehl-Neelsen staining are the example for differential staining. In Gram staining, we can differentiate whether it is Gram positive or Gram negative organism. The Ziehl-Neelsen staining is done for acid-fast bacilli or acid-fast organism. By the Ziehl-Neelsen staining, we can differentiate acid-fast organism or non-acid-fast organism. And this special staining, we can demonstrate structure of bacteria like spores, granule, or capsule. That means with the help of this special staining, we can demonstrate the specific structure of bacteria like spores, granules, capsule, or flagellas. And the examples of special stainings are negative staining, flagellar staining, and spore staining. Simple staining. Simple staining, we use a single basic dye. Examples, methylene blue, carbol fuchsin, crystal violet, and saffron. Saffron, these are the basic dye used for simple staining. And this simple staining is a single step procedure, we can say. And here we are using only single stain or dye and also a single step procedure. Just we will make a smear. After that, we will add this basic dye such as methylene blue, carbol fuchsin, crystal violet, or saffron, and just we will wash and we'll observe under the microscope so we can see the shape and arrangement of the bacteria in simple staining. Differential staining. Differential stainings are, we can differentiate bacteria into two. Examples are gram staining. In this gram stain, we can differentiate whether it is gram positive or gram negative organism. And acid fast staining, we can differentiate whether the organism is acid fast or non-acid fast organism. So these are the examples. Gram staining and acid fast staining are the examples of differential staining. Gram stain. Danish bacteriologist Hans Christian Grams, who first introduced in 1882. That means the gram stain, the name grams, that came from this Danish bacteriologist Hans Christian Gram. He introduced the gram staining procedure for bacteria. And this gram staining is an example for differential staining. That means in differential staining, we can differentiate bacteria into two. In the case of gram staining, we can differentiate the bacteria into gram positive organism or gram negative organism. Gram staining procedure. The gram staining procedure involves four steps. First step is the primary stain with crystal violet. Second is the modern iodine. Third step, dechlorizer, that is alcohol. And the last and fourth step, the counter stain, we use saffron. In first step, to the smeared slide, we will add crystal violet. So this crystal violet will go and bind to the bacterial cells. So here you can see the organism will appear in violet color. And after adding with this crystal violet, we will wait for 1 minute. 1 minute we will wait. And after 1 minute, we will wash with the water. And next step, we will add this iodine. Iodine is a mordant. And while adding this iodine, this will leads to the formation of crystal violet iodine complex. So here you can see all the organism will be seen in purple or violet color. And the next step is the decolorization step. Decolorization, here we will use acetone or alcohol, and the decolorization step is the crucial step. In the decolorizer step, we will add this alcohol or acetone and we will wait for 5 to 10 seconds, and immediately after 5 to 10 seconds we will wash with water. And during decolorization, what will happen? The gram positive organism will resist decolorization with this alcohol or acetone, and the gram negative organism will be washed off. The stain which is attached to the gram negative organism will be washed off by this decolorization step. And the last step, after decolorization, we will add this saffron, and saffron is the counter stain here we used. So what will happen? This gram negative organism will takes the counter stain color of this saffron, that is red color, and the gram positive organism will seen in violet color. And this is the procedure for gram staining. And in this observation, we can see, if you are observing the organism into oil immersion microscope, we can differentiate the gram positive and gram negative organism like this. That means the gram-positive organism will resist decolorization with this alcohol. So you can see the gram-positive bacteria in violet color. In gram-negative organism, after the decolorization step is over, all the stains will be washed off. So the gram-negative organism will take up the color of the counter staining. So it will appear in red color. So the gram-positive organism will observe in violet color and the gram-negative organism will appear in red color. Gram stain principles. The theory behind the gram stain principles are the presence of thick peptidoglycan layer in gram-positive organism, acidic protoplasm in gram-positive organism, lipid layer which is present in gram-negative organism, and more number of pores in gram-negative organism. Coming to the thick peptidoglycan layer, and this is the bacterial cell wall of gram-positive organism and gram-negative organism. Here you can see this is the peptidoglycan layer of gram-positive organism. The gram-positive organism is having thick peptidoglycan layer when comparing to this gram-negative organism. So the stains, the crystal violet iodine complex will come and bind to this peptidoglycan layer and it will resist decolorization with alcohol. And coming to the acidic protoplasm, the gram-positive organism is more acidic protoplasm when comparing to this gram-negative organism. So this iodine crystal violet complex will bind to this acidic protoplasm because they are basics dyes, it will go and bind to the acidic protoplasm. So it will resist decolorization with the acetone or alcohol. And in the case of gram-negative organism, the outermost layer we have a lipid bilayer. And due to the presence of the lipid bilayer, this iodine or this crystal violet will come and bind to this outermost layer, lipid bilayer. So it will not. So this lipid bilayer will prevent the entry of stain into the peptidoglycan layer. So after the decolorization, along with this lipid bilayer, the stain will be washed off. And here you can see the number of pores are more or pores are seen in gram-negative organism. And also after decolorization, patterned stain is bind to this peptidoglycan layer. Through these pores, the stains will be washed off during decolorization. And these are the main principles of gram stain. Acid fast stain. Acid fast stain or acid fast staining is an example for differential staining. So we told that differential staining means after staining, if you are observing under microscope, we can differentiate organism into two. In acid fast staining, we can differentiate acid fast organism and non-acid fast organism. In that acid fast staining, Ziehl-Neelsen method is the widely popular method used to detect or demonstrate acid fast on non-acid fast organism. And the Ziehl-Neelsen method was invented or introduced by two German scientists called Franz Ziehl and Adolf Neelsen. So this by the Ziehl-Neelsen method, we can identify acid-fast organism, mainly Mycobacterium tuberculosis and Nocardia species. Acid-fast staining, Ziehl-Neelsen method procedure. In this Ziehl-Neelsen method, first we will add the primary stain, that is strong carbonyl fission, to the smear, and after that we will heat the smear until the vapors are arising. So what will happen during this heating of this smear along with this stain, that is strong carbonyl fission, the bacterial cell wall is generally thick in the Mycobacterium tuberculosis and other acid-fast bacilli. So while heating, the cells of this organism, the bacterial cell wall of this organism will become thin so that the stain will easily enters into the bacterial cell wall. And after that, we will decolorize the stain with sulfuric acid or acid alcohol. And depending upon the organism, this percentage of the sulfuric acid will be different. Here you can see in this Mycobacterium tuberculosis, we use 20% of sulfuric acid. In Mycobacterium leprae, we use 5% of sulfuric acid. And Nocardia species, we use 1% of sulfuric acid. And acid-fast parasite like Cryptosporidium, Cyclospora, and Microsporidia species, we use 1% sulfuric acid. And in bacterial spore, we use 0.25 to 0.5% of sulfuric acid. That means depending upon the organisms, the percentage of sulfuric acid or the decolorizer will be vary. And the last step is the counter stain with the methylene blue. And we will add this counter stain methylene blue. And after that, we will observe the organism under oil immersion microscope. So the first step is the primary stain. We use strong carbon fusion. And after adding this strong carbon fusion, we will heat the slide until the vapors are arising from the slide. So by seeing these vapors, we can identify that the smear or is already heated well. And we will add decolorizer, that is sulfuric acid. Depending upon the organism, it will vary. And after the decolorization step is over, we will add counter stain with the methylene blue. And the peculiarity of this acid-fast organism is this acid-fast organism will resist decolorization with this acid alcohol or sulfuric acid. So the acid-fast organism will be seen in red color, that color of carbon fusion, and the other acid-fast, non-acid-fast organism will appear in blue color. That will take the color of this methylene blue. So this is the acid-fast staining of Mycobacterium tuberculosis. Here we use 20% of sulfuric acid as decolorizer agent. So this is the pink or red colored rod-shaped bacteria in blue background. This is the sputum smear. So the red color bacilli can be seen in blue background. That is the red color is the color of strong carbon fusion, and this blue color is the color of methylene blue. And this is the acid-fast staining of Nocardia species. The Nocardia species are long or branched filamentous-like organism. So in Nocardia species, we told that we used 1% sulfuric acid as decolorizer. So here you can see this is the red colored filamentous bacteria in blue background. So this is the acid-fast staining of Mycobacterial leprae, and you can see this red colored bacilli, that is intracellular bacilli in the red color in blue background. We can see that is the red color is the color of strong carbon fusion, and blue color is the color of methylene blue. In Mycobacterial leprae, we use 5% sulfuric acid as decolorizing agent. Acid-fast stain principle. Mycobacteria and Nocardia do not stain with simple stain. So these organisms can be stained by heating them with carbon fission. The heating helps the stain to penetrate to the cells. Once the organism has taken up the carbon fission, they are resist decolorization by acid alcohol, that is sulfuric acid. That is why it is called as acid-fast bacteria. And this acid fastness is due to the high lipid content, that is mycolic acid, in the cell wall of this organism. So the main principle of acid-fast organism is the presence of mycolic acid in the bacterial cell wall, and this is called acid-fast bacilli because it will resist decolorization with the acid alcohol or sulfuric acid. Once it has taken the color of primary staining, that is strong carbon fission. And we told that the carbon fusion we will heat until the vapors arising from the slide. So during heating, the carbon fusion will penetrate into the cell. It will trap into the mycolic acid. The mycolic acid is present. It will go and bind to the mycolic acid. Once it is stained with this strong carbon fusion, that is primary staining, it will resist decolorization with the acid alcohol, that is sulfuric acid. That is why it is called as acid fast bacilli, and the main principle behind this acid fastness is due to the presence of mycolic acid in the bacterial cell wall. Albert Stain. Frederick von Albert developed Albert stain in 1884. This staining technique used to identify Corynebacterium diphtheria, a bacteria that causes diphtheria infection, and diphtheria infection is mainly seen in children. And they stain metachromatic granules known as volatile granules or food granules, which is seen in Corynebacterium diphtheria. Albert stain, we stain the volatile or metachromatic granules or Corynebacterium diphtheria. For this, for Albert stain, we used two types of staining, that is Albert 1 and Albert 2. The Albert 1 contained toluidine blue and malachite green. The Albert 2 contain Albert iodine, or Albert 2 is also called as Albert iodine. Albert stain procedure. First we will take the smear, the slide. To that we will add Albert 1. That Albert 1 contain toluidine blue and malachite green, and we will leave it for 5 minutes. And after 5 minutes we will wash the slide with tap water, and after that we will add, that is Albert iodine. Albert iodine means Albert stain 2, and again we will wait for 1 minute. After 1 minute we will wash with water, and after that we will dry the slide, and we will observe the smeared slide under oil immersion microscope. So we will get an image like that. This is the observation of Corynebacterium diphtheria in Albert stain. Here you can see the green colored vegetative cells of Corynebacterium diphtheria, and in the terminal portion here you can see the bluish black colored metachromatic granules in cuneiform pattern. Cuneiform pattern means Chinese letter pattern, that is X and V like pattern. We can observe the organism like this. So this is the observation of Albert stain smear of Corynebacterium diphtheria. The green colored bacilli in the terminal portion, we can see the bluish black colored metachromatic granules in cuneiform pattern. Schiffer-Fulton Microbiologist Eliot P. Schiffer and MacDonald Fulton discovered endospore staining in 1913s, and this method is known as Schiffer-Fulton stain. Here we use the malachite green dye to identify the presence of endospore in bacteria. Schiffer-Fulton stain, we stain the spores of bacteria. That is, spores are the resting phase of bacteria. The spore-related organisms are Clostridium tetani and Bacillus subtilis. We can do this Schiffer-Fulton stain so that we can observe the presence of endospore in this bacteria. And spores have the tough outer covering called keratin. And in Schiffer-Fulton method, a primary stain, malachite green, is applied by steaming. While steaming, what will happen? Outermost layer, that is the keratin layer, will become thin. The spores will enter into the cortex, and this cortex will consist of peptidoglycan layer. So this malachite green will go and bind to the cortex or the peptidoglycan layer which is present in the cortex. So we will observe the spores in spores color will be an green color with the vegetative cell are counterstained with the saffron. So we can observe the vegetative cells in red color with a malachite green or green colored spores here. So this is the procedure for FultonsStain. So for the first step is the application of malachite green to the smeared slide, and after that we will heat the slide. While heating, what will happen? This outermost layer of the endospore will become thinner, so easily the stain will enters into the endospore. Once it enters, we will use the declarator as the water. Just we will wash with water so that this endospore will resist declaration with the water. Next, we will add the counter stain, that is safranin. And after adding the safranin, again we will keep it for 1 minute, and we will wash the slide with water. After drying, we will observe the smear under oil immersion microscope. So we will get an observation like this. The Schiffer Fulton stain of Bacillus subtilis. Here you can see the green colored endospore with red colored bacilli. The red color is the stain of safranin and green color is the stain of malachite green. That means once the organism takes up the malachite green, it resists decolorization with water. So the endospore will seen in green color with red colored bacilli here. Negative staining, we observe the bacterial capsule without disturbing the cellular morphology of the bacteria. In negative staining, we can observe the clear halo capsule around the bacteria against dark background. Here you can see this colorless halo is the bacterial capsule in dark background. That means in negative staining, the stain will not bind the bacterial capsule. The negative staining requires the use of acids called Indian or nigrosin, and India ink or nigrosins are the stain used for capsular staining, that is negative staining, and they are negatively charged chromogen will not stain the capsule because the negative charges on the surface of the bacteria. The bacteria surface is having a negative charge. So this acidic Indian or nigrosin will not go and bind to the capsule of the bacterium, so that we will get an observation like this. That means a clear halo capsule in dark background. So this is the procedure for negative staining. First, mix the bacteria and stain. That means we will take the bacteria in slide and we will add 1 or 2 drops of negative staining, that is Indian ink or nigrosin, and we will mix it well and we will spread the mixture. And after spreading the mixture, we will observe the bacteria under microscope. So we will get an observation like this. That means a clear halo or unstained the bacterial capsule with dark background. Impregnation method. This impregnation method is a special type of staining method mainly to detect the bacterial or to observe the bacterial filament or the fimbriae, that is the locomotory agent for bacteria, because the microorganisms are too thin to see under ordinary microscope. Not only this microorganism, also we know the microorganism are very small. This measured in micron, and we can imagine how thin will be the microbial fibri or the locomotory organ like flagella. So in impregnation method, mainly the silver impregnation method, we use to stain the bacterial flagella. By this silver impregnation method, it will increase the thickness of the structure called, that is the fimbriae or the flagella, so that we can demonstrate the fimbria very easily when comparing to the other staining methods. So this impregnation method we used to demonstrate the spirochetes and bacterial flagella. The spirochetes are slender bacteria or spiral shaped bacteria, and this impregnation method we can demonstrate the presence of spirochetes, and also the bacterial flagella. And this is the microscopic image of silver impregnation method of flagellate bacteria. Here you can see the peritrichous flagella. The peritrichous flagella means the flagella can be seen all around the surface of the bacteria. In all the area, we can see the flagella here. This is the picture of silver impregnated bacterial smear in microscope.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.